Hello to the recording people. Hello to the YouTube people. We are up and running once again. This is the last in a series of 6 of our live classes. So we are going to be, oh, did I change the link? There's a good chance that I didn't change the link. So let me just double check that I have done that for my students in the course. Here's me thinking, oh, I'm nice and early. I was not expecting to be so well prepared for today, but I have no attendees from my students, which makes me think I have probably basically not updated the link for them to get in there. So bear with me for just a second whilst I do that. Luckily, I have everything nice and accessible, so it should just be copy paste 3 times and we're away. Whilst if you're on YouTube, just occupy yourselves just for a little hot second here. Yep, that's definitely yesterday's link. Save. AQA updated. OCR updated. And EdXL updated. Okay, so yeah. Anyone? Oh, no one's actually telling me in YouTube that they were unable to get into their little. Okay, and now I have people joining me in the Zoom room. So well done, Rich. Okay, so final of our series of 6. I am due to get a scheduled power cut any moment now. So I'm just going to state for the record that should I lose connectivity, I've connected to my tethering hotspot, so I shouldn't be affected. My Mac is fully charged. I'm going to lose my second monitor at some stage. So I'll just have to make a readjustment when that happens. If I do get disconnected, I will, what will I do? If I get disconnected, I won't come back to YouTube, I don't think. But what I'll do, I'll log back into the Zoom room for my students and I'll answer just a Q&A today, just on my phone. And then I'll repeat the live class at some point, and then that can go onto YouTube, depending on when we get cut off. If we've done most of an hour, then I'll just call it a day. But I'll let you know in the chat on YouTube, and I will rejoin Zoom if I do get cut off on my phone, just to let people know exactly what's happening there. Okay, so before I say goodbye to YouTube for the time being, any questions there? Exactly. Well, you're answering questions for me there. Who's that? Amritpal? Yeah, awesome. Yeah, YouTube, we're just streaming to YouTube to give students an idea of like what our live classes are like. If you want to ask questions, then go through tt.co.uk, well, not tt.co.uk, tailoredtutors.co.uk. You can join in the free trial there and then find the live classes once you're inside one of the courses. We do weekly live classes for all our members, but during Easter, we've put on some extra lives to do. I've been focusing on exam technique, so check out the recordings of those past live recordings. They are all on YouTube as well. There's a lot of gold in there. Like if you are struggling with your biology exam technique, make sure you watch them and you practice those skills. Just watching the video is not enough on its own. You do need to obviously practice those things to make sure that you are, yeah, you can't learn it all perfectly the first time. Okay, which of my students in the Zoom room have done or had a look at, if maybe not completed, the question paper? Hi, Kia. Who's had a look at the question paper that I put up? Obviously, you guys are pretty busy right now. You've got revision coming together. You're maybe scrabbling to finish some content. You're trying to maybe join the TT live classes. So there's lots of, so Kia has done some of it, but not all of it. I asked my students to basically apply and practice the exam technique that we have been working on over the last 6 sessions, or the last 5 sessions, I suppose. Everyone else can pull up the chat, say hello to me in the Zoom chat. You have the question paper. Maybe I'll just screen share that before we get too distracted, just in case you want to see where that is inside your the biology live classes. You've obviously got the recordings of all of last week's lives, and you've got, oh, I haven't done yesterday's recording. I will update that this afternoon. And you've got the question paper for today's live class available here. I will make the mark scheme available when I add the recording. For right now, we're going to go through, obviously, those questions together. So Kia, were there any tricky things in there? Any questions? Anyone who has looked at the question paper that I put together, what were any tricky questions? I don't necessarily have to start at the beginning. There's not that many questions in here. Everyone's a bit asleep, I think, today. I suppose it feels a bit like a Friday, but it's definitely a Thursday today. Okay, I'm going to change up. I'm just going to wake everyone up. I want everyone engaging in the chat. You can do this on YouTube. You can do this in Zoom. Set your chat settings, panelists and attendees. If you're in the live, for you, and this is going to be an individual personal response for you, what do you think your biggest obstacle is to getting more marks on biology papers? Where do you drop most of your marks? Where do you think that is? And I'm going to leave that as a totally open question. I'm going to just see what you put. We've been doing lots of work on this, and if you've been in my live classes for the last sort of 6 weeks, then we've been I've been trying to build like what's called a dichotomous key, and it's actually very difficult for, it's a bit nebulous right now, but I'm getting closer to a perfect solution. So I can see you all in here, people. Amy, Ariba, we've got Hashini, Imran, Itaza, Jamie, Jasmina, Kia, Lucy, Marima. I've only done A to M there. Let me know, where do you drop most biology marks? What do you attribute that to? Not identifying the actual topic of the question, not always doing a self-check, but this week has helped in refining that. Thank you, Kia. Data analysis and evaluate questions, as well as running out of time. Excellent. Math skills from Kia as well, the practical skills. Shanti, can you narrow that down, the practical skills? Is it like often there's a stats element to that? Is it the question analysis element to that? Obviously, practical skills is kind of like a topic there. Practicing the practical skills questions and not just getting stuck into your logbook and just going over the experiment that you did. Remember, your exam board don't know which exact experiment you did. If it's an enzyme catalyzed reaction, can be any enzyme catalyzed reaction, and therefore you need to look at the principles and the theory behind it, not just, oh, I did catalase and liver, and that's the one that I need to learn in minute detail. That's not the right way to go about it. Identifying the topic of some questions, not using enough key terms from Jasmina, the math questions, data analysis, practical skills, the analysis, rates and ratios and time management. Yeah, and I totally forgot to update the lives page at all yesterday, so I was so busy. I will do it as soon as we finish this lesson. Normally I can't update it. I can't upload today's because it takes a little while to process, but I can upload yesterday's immediately. And I'll also put the mark scheme for the question paper there as well. And then I'll upload the recording a little bit later on. Okay, I've been nattering for 4 minutes now. I'm going to just rearrange my, I'm actually just going to wait for my monitors to switch off, because everything's going to go off when the power goes off here, which could be literally any second. But fingers crossed, I don't get disrupted. If I do get disconnected, I'll rejoin the Zoom room and just do a Q&A on my phone. And yeah, I'll reschedule the live if I have to, if we haven't done very much of it. All right then, eyes down, dropping in to the iPad and the question paper. Did anyone have any specific questions in this question paper in the Zoom room that you definitely want to look at, that you were like, what? I don't get this, or this was really tricky? I'd pick topics that are going to be accessible to everyone, but obviously the types of questions might be a bit tricky. 2.2. Okay. Okay, well then, if you haven't looked at these, and you remember, the purpose of this document is to try and train us to practice our exam technique. Okay, so I'm getting 2.2 and 9.3 and 5.2. I might just write them down because they'll be off my screen in a moment. Yeah, 5.2 and 9.3. Evaluation of chloride. I don't know which one that is off the top of my head. Let's begin with, we need to do 2.2. We're going to need to analyze the graph. So let's drop in straight away for that. A student. What do we know if a student is doing a practical? What does it give us a little bit of opportunity to do? critique their method. They might have ballsed it up. Basically, it's possible. So whereas if it was a professional scientist, you don't generally look for holes in their technique. They investigated, and we're looking, what are they investigated? The effect of surface area on osmosis in cubes of potato. So it's plant tissue. It's a practical. So this is a practical question for those of you who said that you were struggling on practicals. He cut 2 cubes of potato tissue, each with size of 35 millimeters in length. He put 1 cube into a concentrated sucrose solution. So what does this mean? So decoding the question as we go through this, well, let's maybe do that somewhere else. Here we can do, this is a low water potential. Yeah, that's what this means. This is the code here. He cut the other cubes into 8 equal size smaller cubes and put them into a sucrose solution as the same concentration. He recorded the mass at intervals. 1 large cube. So always when we're doing graphs, we want to analyze the graph. We want to look at our units. Are they continuous? What's our units? With time in minutes, and we've got total mass in grams. Everything's okay. We don't start at 0. Just so that you know, just like it doesn't probably make that much difference here, but this is not 0. It might totally screw you over if you're thinking that that is the case. It's worth mapping in your head. Obviously, I can do it in color. We can do blue for this line here, and we can do red for this line here. Just like mentally tagging one line to the other so that you know. Every year, someone will read these questions the wrong way around, and they'll get the wrong thing for the wrong thing. So if you want, you could put large here, small here, do it however you want, but just make sure you are small times 8, I suppose we should have. Okay, we've analyzed the graph. This is the one I chose this question. Okay, well, I know 2.2 is the one, but let's go through this one. We've got describe the method the student would have used to obtain the results, to get the results. Start after the cubes have been cut. Okay. Consider the variables to be controlled. Okay, what's the technique here? Again, I don't want the answer just yet. What's the technique here? We can run through carbs just quickly. Command word, describe, 3 marks. Actual topic of the question is, what is the actual topic of the question? Well, it's a describing method. I mean, like it's not osmosis, right? Osmosis is not going to be a mark here. So it's literally, we are describing the method. Practical skills, maybe. Actual topic doesn't help us out that much here. It's AO3. I think that's a describe, so technically it's AO1. But I'm going to let you make the mistake as opposed to point that one out, actually. Okay. I'm just going to let you do it. I'm going to let you do it. Oh, my phone's over there. I think I can do a timer. Oh, we're at 39. You can have until 42. Go. I'm going to bullet point some answers, but I'm going to pause my screen share. In fact, no, I'm not. I'm going to do the calculation question that's coming up because it's going to take us a whole bunch of time. So I've got 2 minutes. Can I do this in 2 minutes? Probably not, to be honest, because it's a big one. Shit, what's my time interval? 1 minute to go. Oh my days. Okay, I'm not going to give up on that rest of that calculation because I'm not going to finish it in time. Okay, what do we got? What do we got? Send your answers if you've written on paper. Then have you applied the principles of carbs? Have you done the review of the key terms before you thought about this? Obviously, have you bullet pointed your answers? Nice, short, concise sentences. Did you do a self-check to add in more key terms? Maybe highlight the things that you think are going to get the marks. And then if you're missing something, then that's going to reveal it to you. So cubes need to be blotted dry and weighed. Yeah, so we are specifically here. The reason I chose this one is that we are describing the method. So it's like the stages. It's quite rare. Edexcel do this with devise and investigation. Not quite rare for the other examples to do it, but they do. So that is something that we need to do. We are also obviously weighing them at intervals. So we can't just weigh it once, right? Because how do we know? If we weighed it once, we'd have a straight line on this graph. We'd have a start mass and an end mass and a straight line between them. We clearly have measured, I don't know, 20-minute intervals would probably be the most sensible suggestion. There will be some flexibility with what they accept for that, I would imagine. So you're going to repeat take them out the water bath, dry them. You can also think about some limitations. The sucrose level has to be covering the entire all surfaces of the potato so that it's a fair experiment. If it's the potato's half out of the sucrose, then it's not doing osmosis through that surface. We also want to consider variables, which should just be a couple of sentences, same pH, same temperature. We don't have to state why we're keeping those variables the same because there's no explain in this question stem. So we can just state the variables that could affect the rate of osmosis. So basically temperature, pH. We've got sucrose concentration. It's already been controlled. Tear the scale. You can chuck in stuff like that as well. Let's check and see what this here mark scheme has got in common for us. Okay, so a method to ensure the cut surfaces are, okay, so all the surfaces. Why the 8? Why not all surfaces of all pieces of potato are exposed to the solution? So what you wouldn't want, if this is our beaker, you don't ideally want them on the bottom, right? Because then the underside of them isn't being exposed. So that's a tricky thing. You can put in like a gauze and then have them sitting on the gauze is one way of doing it. Again, the point that I was mentioning about if this is our cube of potato, if the sucrose level is here, then all of this surface area is not being exposed to the sucrose. You don't have to specify yet. You could agitate it regularly. You could put in obviously ample sucrose. Basically, they're not determining what method it is, but a method to make sure all the surfaces of all the pieces of potato are surrounded by sucrose all of the time. But you need to give some details of the method. You can't just say, make sure that. You could shake would be fine. You could like make sure they're sitting on a gauze so they're elevated off the bed of the beaker. I can see most people have done that here. Method for controlling temperature. I can see water bath from Anam there. Into a water bath from Kia, 30 degrees for 20 minutes. So you can state the temperature as long as you're saying it's controlled. Drying the cubes before measuring. So blot them dry, I see from Kia. You want this to be reasonably fast. You don't want to let them air dry because then there is evaporating happening and that's changing our mass. So yeah, I think Sadaf, if you say use an excess of sucrose so that all surfaces are exposed to the sucrose, I think that's fine. You're demonstrating that you understand, I think there, yeah. Credit, no explanations required, as I stated, because the command word is describe. and then measuring the mass at stated time intervals. Oh, that's a little bit harsh, but a suggestion, as I said, was use the graph that they've given you to determine what the intervals are. Okay, there weren't many questions about that. You wrote seconds instead of minutes in FFS. Okay, put it in the journal. Yeah. To be honest, I mean, they can't, if you said check it every 5 seconds, oh, I mean, like that's a funny one. If you said check it every 5 seconds instead of every 5 minutes, technically it's not wrong, but it's probably not physically possible to blot and dry. You've got 9 cubes, right? You've got 1 big and 8 small. No human can blot, dry, effectively weigh, return to the sucrose solution 9 cubes in 5 seconds, every 5 seconds. So if I don't know what an examiner would mark on that one, to be perfectly honest. Okay, the one that you all struggled with. Any final questions on that before we move on? I'm not sure I want to do this next question timed because it is one of those ones which is not 3 minutes. I got 3/4 of the way through in 2 minutes, so maybe it's possible, but you have to figure out what you need to do first. And that, it says in the comments, okay, what was that, Lucy? Every 5 minutes with a maximum between 5 minutes and 40 minutes. Yeah, okay, so 5 seconds would have been nil point. I'm sorry. Okay, let's analyze the question and I'll decide what I'm going to do. The loss in mass shown in figure 3, the graph, is due to osmosis. They're telling you that. I mean, we should have been able to figure that out anyway. And it could have been a suggest question. In fact, not really. It's really a AO2. You should know that. Explain why this lost water or why the mass changed because of osmosis. The rate of osmosis between 0 and 40 minutes is faster in B, 8 small cubes, than A. Is the rate of osmosis per millimeter squared of surface area per minute different between A and B during this time? Use appropriate calculations to support your answer. Before we go into it, what do you think we are being marked for here? What do we believe is what are we getting marks for? This is an odd one, and this is like, I've basically, until this came along, I'd not seen a question like this before. It's a sneaky command word. Method of calculation. To some extent, you will get some method marks in there. Calculating the surface area, the change in mass, and the units. The rate. Yeah, essentially, what must be a stage in this question for us to be able to do it? This is a statement. This is not give your opinion. This is use appropriate calculations. So what's the command word here? I mean, use is kind of one. The traditional command word that we're interested in as a biology person is calculate. Because we must use these calculations. You're right, to some extent, the English grammar word is use is the command, but here the biological command word is calculate. So basically we need the rate per, you shouldn't write actually per, we should use negative indices, I'll show you that in a second, per millimeter squared, let's just do it properly, per min is what we need for A and for B, and then we can state, is it the same or is it different? Okay, so how do we do that? How do we begin doing that? Well, I started doing this up above. So we've got one large cube. Let's do this one first. It's easier, slightly. What's the surface area of this cube? Perfect. It's 35 millimeters in length. So it's 35 by 35 for one side of the square. That gives us the surface area. It's a cube and it has 6 sides. So we multiply by 6, giving us a total surface area of 7,350 millimeters squared. Okay, now its starting mass is 62 grams. After 40 minutes, we read up and across. Finishing mass is 57 grams. So its change in mass, 62 minus 57, is 5 grams in 40 minutes. Ronnie, can we keep the chat on biology, please? It's a biology class, not a maths class, please. So we've got our surface area and we've decreased 5 grams in 40 minutes. So now what's that per minute? 5 divided by 40 minutes gives us 0.125 grams per minute. So now we need it grams per minute per millimeter squared. So I divide all of this by our total surface area and we get 1.70 times 10 to the minus 5 grams per millimeter squared per minute. So we have anyone lost? Okay, Lucy, okay. We're looking to find grams changed per millimeter squared per minute. That's our mission. Do you understand how that's our mission based upon the question? That's my first sort of checkpoint. Let's get rid of these. The rate of osmosis per millimeter squared. So rate of osmosis over the first 40 minutes, we need to, it's not a straight line. It's curving slightly. In fact, A is quite a straight line here, but it's not necessarily a straight line. So we're going to take the average rate per minute over the first 40 minutes. Okay, so you understand that we're working out rate per millimeter squared per minute per millimeter squared. Okay, so that's where we work out the total surface area, and I can label these, I suppose. Our total change in mass is 5 grams in 40 minutes. So that is, so our change in mass per minute is 0.125 grams per minute. So this is rate per min. But we need to know rate per min per millimeter squared. That is the gradient, yeah. But it's not necessarily a straight line, Shubnam. So we've averaged the gradient over the 40 minutes. Why do you not? Because a gradient at, whoa, that was a bit of it. A gradient at t equals 40. Let's see if I can just sketch one on here just quickly. That's the rate at minute 40. We're looking to find the average rate over the first 40 minutes. Between 0 and 40 minutes. Not if it was at 40 minutes. I mean, you could do you have to work it out per minute? I mean, the rate, it would be a very unusual unit to give. If we don't do it per minute, then what do we have? We have what we would have to do here. We would have to do, it would be grams per millimeter squared per 40 minutes. I mean, like, that's not an SI unit. You want to, it needs to be in a, it needs to be per minute as a point of comparison. Sure, if you're comparing 40 minutes to 40 minutes, it shouldn't actually make much difference, but it's got to be per minute per hour. That's a rate. That's a standard rate, is grams per minute, something per unit time. Obviously, that unit time could be per 40 minutes, but that's pretty weird. Does that make sense, Kia? Okay. Okay, so that's the calculation, basically. So it's like 2 maths calculate questions all in one. So we've then worked out our rate per minute. We divide that by our total surface area to give us our grams per unit area per minute, which gives us this value here. Do you want to have a go at doing the other cubes on your own and then compare it? Okay, I think that's a good idea. we're looking at total surface area. And I'm going to give you one scaffold on the way there. We've got, this was our cube. Okay, this was our cube. It's 35 millimeters cubed by 35. How many small cubes have we got? 8. And they're equally sized. How do I need to cut my large cube to get 8 small cubes? We need to slice it across. We need to slice it like this. Then I've got 1, 2, 3, 4. And I've got then 1, 2, 3, and then the one in the back corner that we can't see. Yeah. So what does that mean this distance is? That they are expecting you to be able to do on your own, based upon the fact that they've told us this one is 35 millimeters, and this one is 8 equally sized smaller cubes. So that is brain work that they're expecting you to be able to do without any additional help. So I've given you that. And I've just a quick reminder, how many of them are there? You've told me this just a second ago, but some of you are going to forget. There are 8 of them. So that's an extra step that we didn't have to take into account when we were doing this. Okay, I'm going to give you, let me know in the chat when you're done. I'm going to give you until 2 minutes past. So that's 3 and a bit minutes. Off you go. I will put, well, you pretty much know everything you need to know. No, you don't know everything you need to know. I'm going to put that on the screen. And we're trying to calculate, just the calculate. We're not answering the question right now. You're trying to work out rate per millimeter squared per minute is what I want. Grams per millimeter squared per min for group B. Johnny, I can just see the YouTube chat there. We're going to be, this is basically exam skills. It's nearly all AS. There might be a little bit of A-level. I'll let you know at the end if we do get to the A-level stuff. It's mostly exam technique that I'm focusing on here, although this is just a tricky question, right? A bit of a curveball question. Okay, 3 minutes to go. I'm going to pause my screen share and finish off my calculations so I can talk you through it without. Oh, yeah. Okay. Units. Okay, 1 minute to go. Peace, Johnny. Love it. All right. What have we got? What have we got? Adam, you saying you're done? I'm going to resume my share. Okay, I'm going to go through my workings up here. We worked out together that one large cube is 35 by 35, therefore 8 smaller cubes must be 17.5 in there on each side. So 17.5 squared times by 6 faces gives us per cube. Let's put our units in to be clever here. This is millimeters squared. We have 8 cubes, so we have to multiply that by 8 to get 14,700 millimeters squared total. Checkpoint. How are we doing? I asked twice, Sophia, exactly for that reason. How many cubes are there? I left a subtle hint at that, but I didn't totally make it too obvious. But yeah, there are 8 cubes. So we're comparing all 8 versus the big one. So we have to take into account the surface area of all 8. So this is the total surface area of the 8 cubes, 8 small cubes. Yeah, we have this value. Now, how much mass did it lose? We started at 62. This is from the graph, and we ended at 57. So that was a 40-minute span, and we lost 9 grams in 40 minutes. So again, it's to give us a rate per minute. It's not necessarily it's that mandatory, but I think that's how I haven't checked the mark scheme, so we'll find out in a moment. Per minute, we then work 9 divided by 40 gives us 0.225 grams per minute total surface area. But we need to know per millimeter squared. So we need to divide by numbers of millimeters squared. So that divided by our total surface area, I should always put in my units as I'm doing it, gives us, or gives me 1.53 times 10 to the minus 5 grams per millimeter squared per minute. Excellent. Well done, Sadaf. How do we know that it's basically that comes from reading the question here? Who asked me that question? It was Annam. Okay, so we have 2 cubes, 35 millimeters on all sides. One of the cubes is cut into 8 equal size smaller cubes. That is the only clue that we have. So from that, we basically have to be able to do this deduction that we have one. How do I have to cut one large cube to get 8 small cubes? Well, I cut it in half in all 3 planes. And like that step, there isn't, you can practice the visualization of stuff like that. It's a one-off. I've never seen a question ask that before, but that is a logical step that they were expecting you to be able to deduce this from the text. How do you know? Well, basically our units can tell us this, Hashini, that we're looking for grams per millimeter cubed. And in GCSE, you might have written things like this, grams per millimeter squared, if you're familiar with that kind of units. Then this is our fraction, right? So the grams goes on the top and the millimeters goes on the bottom. Or what X, we could obviously, it's X and Y if we want to just make it generic. It's sometimes easy to say this per that, and the per goes on the bottom and the thing goes on the top. Does that make sense to you? It makes sense to me, but that's, my brain's not always, okay, great. Okay, and let's check the mark scheme. And basically, this is a calculate question, is what we were going. So you would just quote this number, and you would quote this number, and you would say, what is it? Well, let's just check the mark scheme. I think we've spent enough time on this question already. We're past the hour. Okay, calculations per millimeter squared for both sets of data. Okay, so they have made this per minute. They've done the same as I have. I mean, technically, I think you could have calculated it per 40 minutes because they were, as long as it was a like-for-like comparison. They haven't given units here, which is very strange. Normally, I would expect to see proper units in here. So anywhere in the range of 1.6 to 1.8 for the large cube and 1.5 to 1.6 times 10 to the minus 5 for the smaller cubes. If both are correct, that's all you need for 3 marks. No words. If one of them is correct, you get 2 marks. If neither are correct, then you can basically get marks for any of these things, which was the calculation of the surface area of the 2 sets of cubes. So the total surface area with the large cube and the total surface area, 14,700 for the smaller cubes. Or the rates, if you'd calculated the rates, which was mass change per minute over the first 40 minutes. Or 1 mark for saying an incorrect rate divided by incorrect surface area. But if you demonstrated that you were trying to do that as an equation, then you get that mark. Was a nasty one. Happy to move on, or any more questions in there? Thank you, Jake. Okay, I'm going to do a super speed one here. Contrast is our command word. Why am I not in blue? Contrast, the processes of facilitated diffusion and active transport. What am I doing? Oh, it's actually quite difficult for 3 marks. Carbs, command word is contrast. So that means differences. Okay, Marima, I was hoping you were going to say that. Can we say similarities? Compare and contrast, similarities and differences. Contrast only, differences only. So obviously we've got brainstorming our key terms here, concentration gradient, protein, carrier protein, we've got ATP, passive, active, down the concentration gradient, up the concentration gradient. These are the relevant key terms. So what would I say? Facilitate diffusion down the concentration gradient, active transport against concentration gradient, active transport uses ATP, passive facilitated diffusion is passive. Facilitated diffusion can use channel proteins, whereas active transport is always carrier proteins. That's 3. They should all be rock solid answers. I'm pretty confident just like dumping that and moving on. Anyone got any others? Let's have a little check in. Okay, so we've got channel or carrier versus carrier, ATP, yes and no, down versus up the concentration gradient. Okay, so it actually wasn't too tricky. When I saw that, I was like, actually, it might be a bit hard. Okay, which question was it here that you wanted to look at? 5.2. So this one coming up. Students investigated the uptake of chloride ions. Oh, we're looking at barley plants again. They love a bit of barley plants. Chloride ions, Cl-. They divided plants into 2 groups and placed the roots in solutions containing radio. Okay, so we've got 2 groups. The roots were placed in radioactive chloride ions. So it just means we can tell which ones are which. I'm not muted. Anyone else having issues? Yeah, I think maybe your headphones or something kicked in, possibly there, Shreya. Group A had a substance that inhibited respiration. This is code. What does this mean? Well, you've got some stored ATP, but no ability to make new respiration. They haven't specified aerobic or anaerobic, so we're assuming all respiration. Therefore, Jasmina, next step, probably no active transport. Exactly. Well done. You are joining the dots. Oh, I'll write active transport. This is exactly what should be going on in your head when you are reading these questions. Like, that's maybe what's happening. B did not have the substance. So B is the control. Students calculated the total amount of chloride. So it's the total amount, that might be useful, absorbed by the plants every 15 minutes. The results are shown in figure 4. total uptake in arbitrary units. We go to 0, we check the scale, there's no logs, it's linear, nothing too scary there. Time, we're in minutes. So sometimes the graph axes don't always match what says in the question stem. Again, nothing scary about the graph. The units here, we've got 15 over 5 squares. So we've got 5 per square if we want to know that. Okay, we're basically good to go. Nothing too scary here. We want to calculate the ratio. Well, let's look at this. We'll go back to the question. Calculate the ratio, command word, the mean rate of uptake in the first hour, or 60 minutes is what it's going to say on the graph, to the rate of uptake in group E. So it's in the first hour. In this, okay, so the first hour versus the second hour for group B plants. Ah, see, Lucy, now this little bit of question analysis here, Lucy, made question 5.3 make sense? Is that what you're just saying? That's the whole point of decoding the question. I'm so happy to hear that. Okay, so we're looking at group B. We're looking at the first 60 minutes, which is the mean rate is going to be, we're going to read up from 60. And we're going to take, well, let's zoom in a bit here. We're pretty much bang on this line. So this is the rate. So our change is 0 to 320, 40, 68, 360. So we have increased 360 in 60 minutes. This is arbitrary units of chloride ions. Okay, that's the first hour. What's the number for the second hour? So we're now doing here, we're starting at this point. So the increase is everything here. And look, we're on half a square here, look. So we can be expected to read to half square accuracy. So we're going to be here. So 20, 40, 60, 70. So this is 470. But we started at 360. So the increase is 470 minus 360, which you could do in your head, but I never do anything in my head because it's way too prone to making mistakes. We have 110. So I hear it's 110 arbitrary units in 60 minutes. So that's going to give us, there are two rates. So let's just transpose those. 110 over 60 and 360 over 60. So first hour I did in blue is 360 over 60 minutes. And the second hour was 110 increase over 60 minutes. Oh, just do HR is fine. Okay, so what do we have? 110 divided. So this is 1.83 recurring AU per minute we've done here. So we want to make sure we're keeping our units the same. And then 360 divided by 60 is 6 AU per minute. So then we're working out the ratio. Ah, Kia, okay, so you've taken the just max value here. Do you understand why you need to take, because for the second hour, we're starting here, and the increase is this much. Does it make sense now that you see that? Okay, now how do we turn this into a ratio? We've done everything we need to do apart from calculating the ratio of mean hour, first hour to the second hour. So what I want here is I'm going to have my ratio dots. And again, I want my first hour here to my second hour here, because that's the way the question has told me. Well, we divide them both by themselves. Or no, we divide them both by the smallest number. So this one divided by itself is going to give me 1. I know that that's going to be 1 because anything divided by itself is 1. But I then also need to divide that by the same value over here. So 6 divided by 1.83 gives me 3.28, which might be a bit much for a... Have they given us any units for our level of significant figures? Whole numbers. This is 3SF, but it's to a whole number. I think 2dp or 3SF is fine. Maybe 3.3 to 1 is also acceptable. They've also given us the to 1 here. So all we need to do... I don't think they can legitimately mark you down for being more accurate. So I would probably go with that as my answer. I got 278. My full number was, okay, 3.278. So if I'm going to round to 2dp, then I need to do it correctly. So 3.27 would not be right. Does that make sense, Shabnam? Because the full value is 3. Oh, actually, technically I didn't use my 1.83 recurring. Yeah, okay, so if I were to use the full value on the calculator, you might be right. So what do I get if I do 6 divided by 110 divided by 60? Yeah, 3.2727. So you are quite right. In which case, yeah, if you do the equation 6 over 110, I'm sure that's mathematically sacrilege to do what I've just done right there, but you get 3.2727. I don't even know the proper nomenclature for that. Double dots, something like that. Okay, so in which case, this answer would probably be wrong. In which case, let's go with 3.3 to 2 decimal places and make it not a problem anymore. Let's take a little look-ski. 3.3 to 1. Correct answer gives 2. If incorrect, allow 1 mark for the rate calculated in the second hour. It's a pretty odd. Why do they give you a mark for calculating the rate in the second hour and not the first hour? Because they're weird. No, it says a correct answer. 3.27, I think, would be accepted. 3.28, as I wrote it initially, probably not. You'd get 1 mark for 3.28 for sure. But if you've done it to 2dp and rounded it incorrectly, which I effectively did by not using the full level of significant figures on my calculator, then yeah, they can mark you down if it's rounded wrong. But 2dp should be fine. It's correct. So how are we doing for time? 20 past. So far, my lights are still on. I'm very surprised. Very happy about it as well, to be honest. Okay, now we are giving reasons for the results in figure 4, i.e. the graph. So let's check back in with our question stem over here. A, how are they absorbing stuff? They're absorbing chloride ions into their roots, bearing in mind that we've got this question up here, which is like comparing facilitated diffusion active transport. It's like definitely pointing us in the right direction. They don't always do that, but this question is helping us out there. So A has got a metabolic inhibitor. It's inhibited respiration. Therefore, they've got no ATP. Therefore, they can't do active transport or very little active transport. It's going to be used up. Any stored ATP will use up quite quickly. So what if I told you, if I asked the question, what was the concentration of chloride ions in the solution? What's the answer? Catherine's just given a massive clue in the chat. That could be a valid question they could ask you. Exactly. what is it reaches equilibrium. So this is at equilibrium, 160 AU. What's the question, Jasmina, that I've missed? I see Kia's answer to your question, but I don't see your question. So post it again and I'll check it out. So group A, facilitated diffusion only, reaches equilibrium after however many minutes, because it's passive. Group B, the difference here, it's faster. So what are the differences? We've got an increased rate. It's faster, so because it's using ATP, and it can go beyond equilibrium to get, because we can go against the concentration gradient. Let's answer I'll finish this question, Jasmine, and I'll come back and make sure that you're understood. And again, I shouldn't have shown you that. What's the technique for these kind of results questions? Short nuggets, fact, not flowing prose where you're just like giving answers and writing about the differences and maybe why there's differences. This is the difference for this reason. And what's the useful word there? So, yeah, that's our connecting word. Like group B has a faster initial rate than A, or maybe because is more useful than so in this example. Because it's using ATP. A is slower. You can definitely word this the other way around if you want to. A is slower because it's diffusion only. A levels off because the concentration reaches equilibrium. B, the uptake is faster because diffusion plus active transport. B, fails to level off because it's going against the concentration gradient. B, the rate slows because there's fewer chloride ions in the external solution. Really, that's not a difference. 5 is like kind of irrelevant to the question that they have asked, but I don't complain when there's additional things on the mark scheme. That's explaining, or does it just say explain the results? Okay, it just says explain the results, so it's valid. If it said explain the difference in the results, that wouldn't be a valid question. Can you say completion instead of equilibrium? I mean, you don't have to use the word equilibrium. You can say reaches the same concentration, but completion, I'd say avoid completion, because unless you state what it means by being complete, then probably not. And then which question 9 was it? 9.3. This is a straight up AO1 question. This is an AQA question. So OCR students, there's going to be one thing. If you have done this question paper, I'm going to just modify the mark scheme because OCR students need to know an additional step here. I'm going to do that at the end, OCR students, because it's not, that mark scheme is not the easiest one to adapt right there. I'm just going to, I'll give you the updated answer for you in a moment. All right. Last one. Scientists measured the translocation. What's going on inside my head? Phloem, active, does use some ATP, up and down. Oh, it told us it's the phloem. It's in a tree. Not that relevant, I don't think, but it's a big plant, not a small plant. They use carbon dioxide with radioactive carbon-14. Again, it's just a carbon that we can identify. It's different to the other carbon. They put a large, clear plastic bag over the leaves of the branches. Again, maybe a visualizing. This is maybe let's make my leaves of my tree green. And the clear plastic bag goes around the branches. And inside, we have our CO2 with carbon-14. A little sketch helps you visualize it. At regular interval, the branches of each tree added. The main trunk of the tree was not in the plastic bag. Okay, so yeah, we're pretty much here. Ground level here, some roots going on. At regular intervals, after adding the carbon-14 CO2, the scientists measured the amount of carbon-14 released from the top and the bottom of the main trunk. Okay, so no. So maybe from here and from here. at regular intervals. So we have our top. Maybe I could do the bottom in yellow. It might make a bit more sense. On the surface of the trunk, okay, I'm a bit lost in my question. After regular intervals, the scientists measure the amount of carbon top and bottom of the trunk of the main trunk of the tree. On the surface of the trunk of these trees, there are pores for gas exchange. Okay, so we can detect, I guess. Okay, so there are some stomata-like things called pores on the trunk of the tree itself. Nice to know. Mean amount of carbon-14 released from the trunk, coming out in arbitrary units, time after the gas was given in hours. So what does that tell us? It tells us it peaks here before it peaks here. So it's moving down the trunk, which is kind of what you'd imagine because the source of the CO2 is at the top and it's working its way down towards the roots. This is quite a long time scale, 48 hours. Okay, yada, 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 yada. What's happening in this time? How is the CO2? Oh, no, it's actually, what's the CO2 doing here? This is a side question. What's the CO2 doing here? So we've got CO2 or carbon-14 CO2 in the bag up here. Okay, so it goes through the light independent reaction or Calvin cycle, gets made into useful organic compounds. I'm going to call it glucose for the sake of it, but it could be other compounds as well. In fact, let's call it sucrose. It makes more sense as sucrose. It then gets moved around. How? By translocation. And then it gets respired back into CO2, and then it's released. Not mandatory for this question, but they could ask questions about any point along this chain, basically. Yeah. So that's our general process. What's next? I've just literally given it to you, and I promise you that wasn't a setup. It's just like where my brain goes when what might they be asking questions about it? Okay. So converted in photosynthesis into useful organic compounds and then respired to produce CO2. Do we need to say aerobic respiration? It's a plant. So anaerobic respiration in a plant produces, oh no, it does produce CO2 as well, because we go from pyruvate, to acetate. No, we don't. That's normal Krebs cycle. We go pyruvate into, help me out here, ethanol, and then into ethanol. But this is a 3-carbon compound. This is a 2-carbon compound, so we lose some CO2 no matter how a plant respires. So we can just write respiration. We don't have to state aerobic respiration. But typically it's going to be aerobic respiration. All right, how long did the carbon-14 to get from the top to the bottom? So top of the trunk to the bottom of the trunk. Explain how you reached your answer. So one is the time, and then one is the reason for our 2 marks. This is probably more simple than you think. What are your thoughts here before I tell you the answer? I did kind of highlight it, but that was, yeah. So we can assume that the distance between the peaks is marking the same point. It's quite a clean peak. Exactly. So we've got, well, I've drawn on it, which doesn't help particularly here, but that is what you do here. How do you, I've got asked the question in the past, how do we know? Again, here, the peak is technically mid-square, right? And this one is like 1/4 of a square, but they'll only expect you to read to half square accuracy. So which one is this closest to? It's probably closest to this line, really, rather than, they will accept either the value for, what's going on here? Okay, let's get really in close and personal with this one. I'd say the peak is here, technical peak. I think they will allow either the half square mark or this square mark for that. They're not expecting you to read to a quarter of a square. You can if you want to, but they don't expect you to go beyond half of a small square. So just bring this one down. So halfway here, and either this one or this one. What do we get? So 24. So 5 squares, 1, 2, 3, 4, 5 is 24 hours. So this is 48. So what's 24 hours divided by 5 means 1 square is 4.8 hours. And we've got, where are we at? We're at 2.5 here. So 2.5 times by 4.8, 12 hours plus 48. So this is 60-hour mark. And then what have we got here? 48, 72. Which one do I want to take? I'm probably just going to take it. 8. What did you guys choose for your value for this one here? This peak. What were your choices? You chose the middle, half square for both. That's probably a decent amount here, a decent suggestion. I mean, either should be accepted because, okay, in which case 72. So this is 2.5, 2.5 times 4.8 for each square plus the 72 hours. So that's the 84-hour mark, yeah? So the time delay here, do we need to know the time delay? Yeah, well, it's exactly what we're working out. So 84 minus 60. $24 hours. Explain. Oh, what's the reason? This is obviously signifying it's the time between the peaks. But do we have to give more detail than that? Let me read the question. Do we need to say what the peaks represent? The peaks represent the highest rate of radioactive carbon, respired radioactive carbon. Distance between the peaks shows that, the peaks are clearly identifying the same point in the flow of radioactive carbon. Yeah, it's a tough one to explain. The peaks represent the same point in the movement of radioactive carbon through the trunk. Therefore, the distance between the peaks is a measure of the time taken for carbon to move from top to bottom. Yeah, it's a tough one. Are you with me on that? And then what other piece of information is required to calculate the mean rate of movement down the trunk? I'll just do both of these together and then we know how long it takes, but what don't we know? The distance between the top and the bottom, the length of the trunk, because we need to give it in millimeters, meters, centimeters per hour. would be the classic, right? So we'd work out the length and then divide it by 24, and we'd get the distance moved per hour. Yeah, so here I would state here, the peaks represent the same point in the journey of the radioactive carbon. Therefore, the distance between the peaks tells us the difference between how long it's taken to get there. Oh, I need to remember to... About 30 hours. That's weird. Because we used the half square, which was the biggest possible time interval. I'll double check. I think that's just wrong. I'm pretty confident about that. I will, like, there is time. Oh, it literally just says the time between the peak at the top and the bottom. So it doesn't even give us. Shad, you asked me about this in the exam guide, and you can. I'm pretty sure that I have looked at this before. I was pretty solid on my numbers there. That's definitely half square, which is definitely. Do you all agree with my numbers here? That 60 and 84. I haven't done anything weird with my calculations. If anything, it's less than that time interval. You could definitely have taken it to this point here, which would have been less. It's definitely not here. 100% it's not. Let's erase all my scribbles and just scrutinize the paper. Just extra careful. No, without a doubt. I mean, you obviously don't have a power of zoom in your exam, but like the peak is here, which probably is slightly closer to the half square than the full square, but it's definitely not here. The mark scheme is wrong. And we need to know the length of the trunk. Okay, I'm just going to run through. This is describe the process of translocation for AQA and Edexcel students. If you are OCR, I'm just going to do this on a blank. We have our phloem sieve tube element. We have our end plate here, our end plate here. We have our companion cell here, and then we have a non-companion cell here, probably a mesophyll cell. This is OCR people only. You basically, they include the extra stage that you actively transport protons out of the companion cell. And then you get sucrose. Comes in by co-transport through a co-transport protein to actively load the sucrose into here, which then moves via the plasmodesmata into the phloem to create the low water potential and to suck water in from the xylem. Oh, it's a terrible straight line. In fact, let's choose a different color for the xylem. Let's choose, we'll just choose blue for the xylem. So that's the extra step that OCR students need to know, that this is active transport. Oh, me oh my. And this happens first, and then this happens second. You actively pump protons out, and then the sucrose and the hydrogen ions come into the companion cell. That's how the companion cell gets loaded, and then the companion cell, it moves in here. Whereas you'll notice with other exam boards, they just don't include that level of detail, and they'll simply say that the leaf sugars, they don't even specify sucrose here, actively transported into the phloem. And they're into the phloem, they're not specifying sieve tube element or companion cell here. They're just stating it's the phloem. Oh, okay, by the companion cells is what they're saying, that the companion cells are the bits that do the active stuff, which is true, but the reality is they're pumping the protons out so that the sucrose and the protons come back in again together. This decreases the water potential in the sieve tube elements. Again, OCR students do not use the word sieve tube cell. I have seen them rule out. I use this mark scheme actually to build my exam guide question, my content guide question, and sieve tube cell is not accepted by OCR. It has to be element. This increases the hydrostatic pressure, and then it moves from source to sink. I would like 5 minutes of reflection on the things we've learned in the last 6 weeks. If you were summarizing your top tips to your schoolmates who were not at those live classes, what are your big takeaways? What are the things that you think will actually make a difference? I might ask you the bits that always do carbs. Okay, yeah, wonderful. Question analysis tool and answering tool, command word and marks, the actual topic of the question, reviewing the key terms, bullet point, short, concise answers in bullet points, self-check, can you add more key terms? Bullet point facts, little truth nuggets, little golden little bullets of fact, not flowing sentences. You might be a fantastic writer, but that's not helping us out for A-level biology exam technique, especially those sort of evaluate data questions. Do not use all the answer lines. In fact, try and do a paper where you use half or less. And so basically it's an alternative way of doing the 5 words per mark, right? The reason I choose not to use that is because I would rather more short bullet points. But yeah, don't use all the answer lines. They are not relevant. Check you're using all the key terms possible. Make sure you analyze the question properly. In practice, spend that time analyzing the question. Lucy had the penny drop when she did, this molecule is a respiratory inhibitor, therefore no ATP, therefore no active transport. Boom, the next question is opened out for you on a plate. Take graph sections at a time, 100%. So break graphs down into straight line sections. Any difference you look at by straight line section. Yeah, read with your questions with your pen, translate information, decoding the question. Why do they become tired? So that's like an aerobic respiration question. Understanding the command words for evaluate, for and against. Yeah. A contrast is differences only. Compare and contrast is similarities and differences. Yeah, you saw a 6-mark question, gave it 2 pages of writing. Try next time, 8 bullet points, Shabnam. 8 bullet points, 8 lines. Okay, in practice, you can get away with like these super concise bullet points. In the real exam, you might just want to elaborate slightly more, but not, you know, like half a page max. Suggest questions of what is it similar to? For evaluate questions, pick apart the data, especially if a student did it. Understand what the general trend is of the data before you start answering the questions. That, again, you're doing the thinking is much quicker than writing. So you want to do the thinking processing in your head so that you can write less, but better stuff. That is going to speed you up. Because if you're writing a full page or 2 pages for every 6-mark question, that is the time pig, right? And you're probably writing that much because you're not clear on what you need to write. And so you keep writing until you've got nothing else to say. And if that's your approach, you're going to run out of time. Don't just jump straight into writing from Amy, 100%. Fatima, I'll come and answer your questions on YouTube just a second there. Yeah, the commonalities for evaluating conclusions of practicals. No stats test, sample size, was it in which species? Like, can you compare across species? And I'm going to macro that point and say, practice questions by type. If you practice 10 evaluate a practical questions in a row, maybe not 10, 5 in a row, you'll be like, oh, hang on a minute. Didn't do a stats test, was on 4 out of the 5 mark schemes. I can just write didn't do a stats test if they didn't do a stats test. I try to show you the things. Don't just become an algorithm and just like follow answers sort of rote style, you have to use your brain in biology exams. Like, there is no way avoiding it. I'm just showing you some of the tools that you can make your life a bit easier with. Know the sentence you're going to write before you write it down. 100%. Again, more quick brain work, less long flowing. You'll use fewer words, you'll save yourself time, you'll be more punchy with your sentences. One fact or one key term per sentence, full stop. If it's more than that, write 2 sentences. Have a clear objective for every single sentence. Self-check, can you fit in more key terms? Wonderful. You have become, how much, arbitrarily on a scale of 1 to 10, do you think your exam technique has improved in the last 2 weeks? Completely subjective question. 11 from, that's nice, Itasa. 8, a lot from Sadaf. 10, 8. Okay, so like that was 100% my objective, was not really to get too bogged down in the content of the questions, but like, yeah, and Kia, you do practice. I've shown you the path, but you are now the ones that need to practice it. It's not going to become second nature by watching me do it on YouTube or watching me do it in a live. Now, take those tools, practice them, screw them up, get them wrong, like in your past papers. But you're gonna learn. Also, you'll identify the questions like, oh, carbs actually isn't going to be that useful to me on this question. I'm not going to spend the 7 seconds it takes to run through all those key terms in my head, because I'm describing a method, the describe a method question. The carbs not that useful for that one, to be honest. So, and you'll learn that, but with practice. I don't have a perfect way of deciding whether carbs is relevant or not. Definitely for AO1, describe questions, and most AO2, and quite a lot of AO3. And use the Markside drop sheets with the TT revision method, and make sure that you don't just live in content land. Most of the marks that students drop, and I've done this through the poll questions, most of the marks that students drop are because of not understanding the question, or not using the right exam technique or answering technique. And if you're only revising content, then you're not actively working on the things where most potential gain is in the technique. You can use the knowledge that you've got most of the time. Maybe another exercise is just take a mark scheme without looking at the paper and just put a tick, regardless of what the question is. Do you know that statement or not? You can put a ticker across. Know this point, don't know this point. You'll find most of the mark schemes, you will know most of the points, which tells you you don't need more content knowledge to get more marks. Some of it, yeah, some of it you do need to know more, but most of the time it's not that you're lacking content knowledge. Yeah. Amy, I just used to focus on content when revising. You're just living in the exam guide now, Kia. These are all making me very happy. This is telling me that you will improve more. If you only use the content guide, then you're going to get better. There's lots of exam questions in there now, but like this is what I want to see from you. Okay, wonderful. Jasmina, are you allowed to ask explanations 2019? You can, if you send me a question from the 2019 in the comments, I can answer it. I can go through bits of it with you for sure. Yes, I can. I'm just not allowed to give you the questions. All right, Fatima on YouTube saying, how do we have access to the notes? Through our academy. So all the recordings of these live lessons, all of my template notes, all of my content guide lessons covering the entire course, math skills, practical skills, everything is inside the Tailored Tutors Academy. So tailoredtutors.co.uk. If you've never tried us before, do the free trial. If you have tried us before, then you know what we're like. So you can just go to the pricing page and sign up today. In your school, you have 5 mini assessments: biological molecules, cells, proteins, digestion, transport. Okay, you're just stating that. Okay, no question there. Any final questions? Next week, we are returning to the normal live class time, Tuesday, 5:30 p.m. there were quite a lot of questions submitted for yesterday's class. I put some of them in the document today. Some of those that I didn't get round to because there were too many questions, I will cover on Tuesday next week. And so again, as always, check out the live classes page. I'll update yesterday's recording in the next 10 minutes here. I'll put the mark scheme up for today in the next 10 minutes when I update those pages. And I will update the recording for today later tonight when it's processed. Are you allowed to ask about assessment questions that we have access to, but not the mark schemes? Yeah, you can ask me questions on absolutely anything you like in the course. I'm there as your tutor to answer them for you. Response to stimuli. What I'll probably do is I think I'm going to just take control of the next live class. I'll probably gather opinions as to what is the best use of our time moving forward between now and the end of the year. Seeing lots of gratitude and improvement going on. And parting message is TT revision method, practicing this technique is the way now for all of you. It includes topic revision. If you know that, if it shows you that topic content revision is what you need to do, then you can do topic content revision, but don't do any topic revision without doing a question and the principal reason for you not getting marks being that you didn't know the content, which does happen sometimes. But be aware that it might be you didn't understand the question. It might be that you did know the marking points and you just didn't access them for whatever reason. I believe in you all. Good luck with all your assessments. Ask me questions inside the course. As always, I'll be there. We'll be doing 24 hours. I don't think a single question has gone more than 24 hours, apart from Alex, who's asked me a really one that I need to sit down and think about, and I've just been too busy to think about. Alex, if you're there, I will do that this week. That's my commitment to you. See you all on Tuesday. Oh, what have I done? I've moved everything off the wrong place. Oh, my days. I can't even find the buttons. Okay, here we go. So that's goodbye to YouTube. I'll check the chat in just a second just to make sure I've not left any unanswered questions. And goodbye to the Zoom.